conference is now being recorded.
Welcome to the AMDA webinar, Spark Therapeutics Gene Therapy Research. Your speakers for today's program are Federico Mingozzi, PhD, Paul Monahan, MD, and Amy Fisher, MS, CGC. Audiocast quality is subject to your equipment, available bandwidth, and internet traffic. If you experience satisfactory audio, please use the telephone dial-in option provided in your confirmation and reminder emails. If you have dialed in, operator assistance is available by pressing #. You can send questions at any time by using the Q&A box located on lower left of the presentation screen. You can type your message at the bottom of the Q&A box and click the Ask button with your mouse to send it. I will now turn the call over to the speakers. Please begin.
Hello, everyone, and welcome. Thank you so much to the AMDA for affording us this opportunity to share with you all an introduction to Spark Therapeutics and an overview of gene therapy research. I'm Amy Fisher. I'm one of the patient advocacy leads here at Spark, and I know that many of you are very familiar with industry and know that we work in a very regulated environment, and we have many rules in place within that environment for very good reasons, but those rules do govern who and what and how we communicate about our work. And so there will be limitations to some of the questions that we can answer today, but we are very excited to provide you that introduction to Spark, share a little bit more about who we are, where we work, and in what areas that we work, and then take the time to overview gene therapy. We've learned from our experience in a variety of different therapeutic areas that this type of content is really of great interest. And I think any time that we have an opportunity to gather together and discuss a little bit more and ensure that we are all grounded in the same understanding of the science, we are positioning ourselves well for ongoing discussion. So thank you again very much for taking the time to spend a few minutes with us today. And I'll now turn it over to Dr. Mingozzi.
Hello, everyone. And again, I'm very happy to have the chance today to discuss and present Spark to all of you. I will start by the next slide, which shows a little bit of the history of Spark. So Spark is a fully integrated company committed to discovering, developing, and delivering gene therapies. What it means fully integrated? It means that in our organization we have all the functions that are needed to bring gene therapies all the way to patients. This includes research laboratories, the functions that are involved in clinical development, regulatory, and manufacturing, and they're all part of our organization. Spark is a company that was founded 5 years ago in March 2013, and it originated from the technology and knowledge that was gained over decades of work done at the Children's Hospital of Philadelphia. In fact, it originated from a research group led by Catherine High, which really worked on gene therapy and developing the technology and translating that to patients. The organization includes scientists and clinicians. A lot of us are people with a long history of work in investigational therapies, which include gene therapy, and really a lot of our work was devoted to the validation of the technology to explore the potential to provide long-lasting. In the next slide, you will see us three today, the people who will be presenting at this webinar. So I'm Federico Mingozzi. As I say, I'm the Chief Scientific Officer at Spark. I joined the company about a year ago, and my history comes actually from the Children's Hospital of Philadelphia, where I started as a postdoc working on gene therapy and working at the translation of the early work into the clinical trials for hemophilia and other genetic diseases. Then in 2013, I actually moved to France to Geneton, a non-profit research organization owned by the French Muscular Dystrophy Association, and there is when I started to work on POMPE disease in the lab. We started to do research to develop a gene therapy for POMPE. And then, as I said, about a year ago, I moved to Spark at the same time when we established the research activities on POMPE disease gene therapy. Then I leave the next introduction to Paul.
So I'm Paul Monahan. I'm a pediatrician, a hematologist oncologist, and I worked at the University of North Carolina for about 25 years, including work on gene therapy for hemophilia and other attempts to deliver genes for gene correction to the liver, and have been with Spark since the spring of this year.
And hi, everyone, again. My name is Amy Fisher. I'm one of the patient advocacy leads here at Spark. I've been involved in rare disease for almost 2 decades. I got my start as a clinical genetic counselor, also at the University of North Carolina, where I was able to focus on the care and genetic counseling of patients and families impacted by lysosomal storage disease. After my time there, I transitioned to industry and then joined Spark just about 2.5 years ago.
Okay, so going again a little bit farther into who is Spark, I would like to just state our mission and vision. Our mission is to challenge inevitability of genetic disease by striving to discover, develop, and deliver treatments in ways unimaginable until now. And the way we do it is through gene therapy. Our vision is a world where no life is limited by genetic disease. And here you see how our research programs are organized. We focus on a number of rare genetic diseases, and the areas are the one you see in this slide. One is inherited retinal diseases, and I will provide you some more detail in the next slide. And then we have a second disease area, which are the metabolic and hematologic diseases, so liver-directed gene transfer. That's how we categorize this area. And finally, we have active investigation in the area of neurodegenerative diseases. Spark pipeline, like in other organization, is arranged in a number of programs that are in a different stage of development. In the preclinical development, so we have a number of programs on, as I mentioned, inherited retinal diseases, which include one of them is hereditary optic neuropathy. And then in the area of metabolic and hematologic diseases, you can see how there's our program on gene therapy for POMPE disease. Finally, as I mentioned, in the neurodegenerative diseases, we have a lysosomal storage disease, Batten disease, and Huntington. Then proceeding, and this really gives you an idea on the variety of the programs we have, and the fact that we at Spark, we have a long-standing expertise in translating research programs to clinical trials and also all the way to registration. So in early phase clinical trials, we have 2 investigational gene therapies, one in the area of inherited retinal diseases, so gene therapy for choroideremia, and then in the metabolic and hematologic diseases, we have an investigational gene therapy for hemophilia A. We also have recently transitioned one of our programs on investigational gene therapy for hemophilia B. Again, is a liver-directed gene therapy to our collaborator Pfizer for the initiation of phase 3 trial. And finally, we are the first biotech company in the United States that really achieved the goal of having the approval for gene therapy for genetic disease, and this is a genetic disease for an inherited retinal disorder. So I think Spark, again, you have, I hope I give you, although quick overview of our pipeline, how we have different programs that are in early stage development or late stage. I think this is the result of the hard work of many people who are committed to the mission and vision of Spark, and who are also working on developing a gene therapy for many diseases, including POMPE. And the way we do that is through our expertise in designing gene therapy vector, in manufacturing them, and our approach to the development of these drugs, also in terms of the way we approach the regulatory strategies when we want to bring them to the clinic. And of course, the most important value we have at Spark is our strong commitment to improve the patient care. So I'll pass the now to Paul Monahan, who's going to talk about gene therapy.
Thank you, Federico. So as Dr. Mingozzi has introduced, the goal of gene therapy, or our work here at Spark, is that we develop treatments for diseases that occur when an individual is born, that's to say, inherits a gene that causes disease. When we talk about a gene, we're talking about a sequence of DNA. And a gene normally provides direction for a normal function of the body, but if the gene information that's been inherited has a mutation or has a mistake in it, then a disease may result, and our goal is to provide therapies for disease-causing gene sequences. There are at least 3 different approaches to try to treat different genetic diseases that are being researched with gene therapy. If we look across all types of diseases that are being addressed, the first in the box to the left shows that one might use gene therapy to introduce a new gene into the body to provide a totally novel function to the body that helps to fight a disease. Another approach in the middle would be to insert a functional copy of the gene, and with the goal here being to circumvent the problem that the normal gene that's been inherited is mutated and causes diseases. So this is gene addition to provide the missing function. A third approach that you can imagine would be that if a gene mutation is causing disease, because not only is the mutation knocked out a normal function, but it's actively causing disease via a new function. One approach gene therapy has been used to is to try to knock out this detrimental gene that may then allow the body to go back to normal functioning. Now, our current metabolic and liver-directed programs all use the second approach here. That is, inserting a normal copy of the gene to restore the body's ability to return to normal function. So some of the language that we use, we talk about gene therapy vectors. We talk about our medications as gene therapy vectors. And I want to give you an idea of what we mean by this. As a vector, we're talking about essentially a delivery vehicle for functional genes. Now, the body, as you can imagine, doesn't really like to just take up any old DNA that it might run into in the environment or might be brought in to the body by various routes. And we actually to be able to provide a functional gene copy, we need to have a delivery vehicle that will take that into the appropriate cell. So you can think of the functional gene as an instruction manual to make a desired protein. The researchers then place the functional gene in a delivery vehicle or vector, and this is most often a virus that's been modified. So one way to think of this is essentially the virus structure is providing an envelope. You're placing instruction manual in that envelope, and then that vector is able to give directions as to where we want those useful instructions to end up. So the vector is intended to work as an envelope addressed to a specific final destination or address inside the body. Viruses, the most prevalently used method for delivery of genetic material in gene therapy research. And this is in part because the natural ability of viruses is to target specific cell types in the body. Over millions of years, viruses evolved to be very good at putting their own genetic information into cells to make the cells, essentially to co-opt the cells to make the virus's protein instead of what the cell was to do. Now, when we make a gene therapy vector, we've got no interest in making any virus proteins. So the viruses are modified before being used in gene therapy by removal of all of the viral genetic information, all of the instruction manual that might come from the virus itself. The viral genetic information is chopped out, is replaced by the functioning gene that we want to deliver to the target tissue. And at this point, we no longer refer to what's been constructed as a virus, similar to the original virus, the parent virus, but instead, what we have here is a viral vector, a gene delivery vector based on a virus capsid shell. So when we leave the outer shell or the capsid intact in the manufacturing process, we place a functional gene instead of any virus genetic material into the vector. The vector then, with the functional gene, the instructions for making a useful and hopefully disease-correcting gene product, that's what gets delivered into the body. The vector is intended to travel to the target cells. Clearly, our programs that we are labeling metabolic and liver-directed are trying to take advantage of many of the natural properties of the liver to act as a place where most of the proteins produced in the body are made. If transferred successfully, the functional gene should provide the correct instructions for the cell to make that functioning protein or stop producing a harmful. And we count now on the body to do this function to provide disease correction rather than counting on repeated intravenous infusions of a protein. So Spark has developed a short film that you may find useful. This was developed in response to questions from the patient community with retinal or eye diseases, but we wanted to get across some of the kind of foundational ideas of how we develop gene therapy vectors and our goals as a company. We're going to run this film. It's about 2 minutes.
Gene therapy research, then and now. The idea of gene therapy is not new. In fact, scientists have been investigating and evolving it for more than 50 years, and to date, more than 2,300 gene therapy clinical trials are planned, ongoing, or have been completed. Gene therapy research, some in very early stages, is focusing on many diseases that are partly or fully caused by genetic mutations, such as blood clotting disorders, for example, hemophilia, cardiovascular disease, neurodegenerative disorders such as Parkinson's disease, vision disorders, and musculoskeletal disorders. The potential of gene therapy research brings hope to millions of people living with currently untreatable diseases. Understanding genetic disease. Before you can understand what gene therapy research is, it's important to know what a gene is. The human body is composed of trillions of cells. Within a cell, there's a nucleus, which contains chromosomes. Chromosomes are made up of DNA, which is the body's hereditary material. Genes are segments of DNA. Genes contain instructions for making proteins, which are molecules that build, regulate, and maintain the body. Sometimes there's a change in a gene's DNA sequence. This is called a mutation and can cause a necessary protein to not work properly or to be missing. A mutation can be a substitution, deletion, or duplication. Some mutations are harmless, but others can result in a genetic disease. Simply put, gene therapy is an investigational approach with the goal of treating or possibly preventing a genetic disease. Exploring the potential of gene therapy. One goal of gene therapy research is to determine whether a new or functional gene can be used to restore the function of or inactivate a mutated gene. One way for this to happen is to deliver a gene into a cell. To do so, a transporter known as a vector is typically used. A vector can be made from an altered virus, which means that before the virus is used, its viral genes are removed. Vectors can be given intravenously, which means they are administered into a vein, or in gene therapy research.
So a few words about Spark and POMPE disease. The Spark Therapeutics POMPE disease program is, as I've mentioned, a metabolic or liver-directed gene therapy research program. The program was in-licensed from Genethon. Genethon, as Dr. Mingozzi had mentioned, is a not-for-profit organization focusing on rare inherited disorders. The gene therapy technology and bioengineering performed at Genethon is then combined with Spark viral vector technology in the therapeutic that we want to take forward. This program builds on Spark's validated platform, that is to say, it builds on Spark's gene therapy vector technology and the manufacturing of gene therapy vectors, and it builds on demonstrated expertise in other areas of liver-directed gene therapy. The program is currently in preclinical development. That is to say that we are completing the due diligence to assure ourselves and the health authorities that the approach justifies evaluation in human clinical studies. And this work is ongoing, both in the laboratory and in the community, to best understand how to address patient and family needs, which is why we're grateful for your participation today, and I'll let Amy Fisher give you some more details.
Thank you so much, both Paul and Federico. And while we've mentioned that Spark is relatively new on POMPE disease, having just announced our program earlier in 2018, we are not new to efforts to work with the rare disease communities that we serve. And what we've highlighted here for you is some examples of that collaboration that we've been so fortunate to have. Fairly recently, NORD put together a really nice Q&A style gene therapy video that was supported by Spark, but informed and guided actually by an individual living with POMPE disease to make sure that appropriate questions about gene therapy were being answered for the community. I understand that some of you may have had trouble seeing that video that we just went through, the Spark gene therapy video. That is available for you on the Spark homepage, which I'll show you in just a moment. And it is accompanied by a brochure. The screenshot of that brochure is up to the top right there. And then lastly, you'll see just a screenshot again of our homepage where we've really tried to develop comprehensive information not only about the science that Spark is conducting, but the work that we have ongoing with the patient and family communities that we serve. And all of these materials come out of conversations that we have with rare disease community members, like the Pompe disease community, building materials that truly aim to address unmet needs or questions that exist about the science that we're conducting and the overall initiatives ongoing in the community. The feedback is sought in a variety of ways and is provided in a variety of ways, including advisory boards and through webinars like today. And so we plan and endeavor to have as strong a collaboration with the Pompe community as we've been fortunate to have with some of our other therapeutic area communities. Now, I mentioned earlier we probably won't be able to address some of the questions that come in today. However, we do have additional resources and available contact information for you. Please do feel free to visit sparktx.com. There is a Pompe section on sparktx.com homepage under the Patients and Families section that you can visit at your leisure. And then the patient email box is monitored by members of the patient advocacy team, and we can continue our dialogue there. Again, thank you so much for this opportunity to speak with you all, and now we can take questions that you choose to submit through the Q&A box.
Thank you very much. Ladies and gentlemen, you can send questions using the Q&A box located on the lower left of the presentation. Type your message at the bottom and click the Ask button with your mouse to send it.
So one of the questions that we've received is, when will your clinical trials be starting? How will we know? At this stage, it's a little bit too early to speculate on exactly the timing of clinical trials. We continue to work through all of the requirements and the diligence in our preclinical experiments and efforts. But you will be assured that when clinical trials are open for Pompe gene therapy with Spark, they will be listed on clinicaltrials.gov, and we also will remain in close touch with advocacy organizations like the AMDA to provide those updates as soon as they're available.
Again, if you'd like to ask a question, please type your message at the bottom of the Q&A box and click on the Ask button with your mouse to send it.
So a question we've just received, what is the biggest hurdle in gene therapy, and why does it seem to be moving so slowly?
That's a fantastic question. I think there are a number of really probably kind of two monumental issues in the development of gene therapy. One has been around the ability, what's involved in being able to get a nucleic acid sequence, to get a gene that may be potentially therapeutic to the right place in the body and have the body take that instruction manual and make the appropriate protein or correct function from that. And I think a lot of the early years of gene therapy, we thought the biggest hurdle was going to be just around can you actually get gene information from outside the body into the appropriate cell and get a protein made. And that continues to be a major focus. It's a major focus of a lot of the bioengineering that was done at Genethon, and why we were interested in licensing that. But the other major hurdle has been, as I mentioned, the body has millions of years of evolution to say that you don't just want to pick up any gene sequences or any nucleic acids that you might run into in the environment. And there are a number of, the immune system is a very complex protective mechanism, and a lot of the work, in particular in the last 14 or 15 years of gene therapy, has been how to be able to deliver the functional gene without upsetting the body and upsetting the immune system to the point where it feels like it needs to raise a major defense. And this has been a real focus of the work here in Philadelphia, and it's been a real focus of the research community. And I think we're in a much better position now of knowing how not to perturb or upset the immune system as we try to correct the disease.
Again, if you'd like to ask a question, go ahead, Amy.
Sorry, I was just going to say a question we received about why a liver-mediated approach to gene therapy.
So this is Federico Mingozzi. We at Spark have a lot of experience in targeting the liver with gene therapy to express protein, and we've done that in the context of our investigational programs for hemophilia. And there's a number of advantages when you target the liver with a gene therapy vector, which really comes from the biology of this organ. The first one is that the liver is an organ that is in close contact with the gut and sees a lot of different foreign entities, what we call antigens, that comes with the food through the gut. And so because of that, is an organ that evolved in a way that it does not reject these new entities, these antigens, when it sees them. Rather, it tends to accept them in a way that when we eat, we don't react to everything we do eat. And so it's a similar concept also when we use gene therapy to transfer to the hepatocytes, the cells of the liver, a gene, and we express a new protein. Then this protein seems to be accepted more easily by the immune system. The other point that makes the liver very interesting for gene therapy is the fact that it is very highly vascularized. So the access of anything that is made by liver cells to the systemic circulation of blood is highly efficient. And finally, the liver is also one of the organs in the body that produces most of the proteins that we find in blood. And so the liver cells are very, very efficient in the production of any protein, including anything that is introduced via gene therapy. So all of this makes this organ very attractive when we want to develop a new gene therapy approach.
Again,
we received another question about retreatment with gene therapy and the thought about whether that would be indicated or not.
Okay, so just a few words. Today in the clinic, gene therapy is intended as a single treatment. And part of the reason why it's intended as a single treatment is because after gene therapy using a viral vector, there's a phenomenon of immunization. So individuals will develop antibodies against the vector, which will make a second administration of the vector challenging. However, having said that, there's a lot of work, including work ongoing at Spark, focused on developing strategies that would allow, if needed, a second or third multiple administrations of vector. And so this is ongoing work.
Okay, we've received a question about vectors and how do we know that AAV is the best approach?
So this is a great question, and I'll say that the person posing the question also followed that up with, is it best when used in central nervous system disease, which I think kind of gets to the point. The AAV has turned out to be a useful vector for going into tissues that are what we call undifferentiated, essentially that are mostly fully mature and tend to have a long lifetime. So especially after the very beginning of childhood, most of the liver is mature tissue. It's turning very quickly and being replaced quickly. Clearly, that's the same with the central nervous system or the brain. It's the nervous system develops and then is relatively stable, and the eye and the retina as well as certain, essentially the retina being an extension of the central nervous system. So the different viral vectors that have been used in gene therapy have advantages and disadvantages. We really are trying to take the approach of what gives us the best risk-benefit ratio? How can we achieve disease correction while also trying to deliver on a promise of safety? The AAV or adeno-associated virus is not associated, the parent virus in the environment is not associated with human disease. So we're starting with a vector that we think gives us a relatively good safety profile, and as compared to using cold viruses or herpes viruses or some other vectors that have been tried in other approaches. The wild type or the parent virus actually can't even on its own go into a cell and cause a infection that can cause lead to the production of more virus and harm the cell. We think that's an advantage as well. One characteristic of the AAV, in addition, is that after entering the cell and expressing our protein of interest or disease-correcting protein, the gene persists in the cell, but not in a form that's inserted or integrated into the host cell's own DNA, which for applications we feel may carry an advantage in that we're not likely to disrupt normal genes within the cell while still getting a long-term function of expression of the gene from the AAV. So that's why we've chosen for the particular organs that we're going into, the particular diseases that we're going into, to try to be able to offer the most benefit at the lowest amount of risk.
Thank you. So we have another question about, has your research included gene therapy in any animal models, which I think allows us to explore the basic tenets of preclinical research?
Yes, so of course gene therapy research and what we call translational research, so all the activities that are needed to bring a therapy from bench to the bedside do require validation in different models. And in the work ongoing of investigation of gene therapy for POMPE disease, a number of models were used, including small animal models or mice. There are models of POMPE disease and also large animal models, which have the advantage, the large animal models, even though there's no large animal model of POMPE disease, they do have the advantage of mimicking the physiology of humans. They're closer to humans. So a number of these models have been used to establish the safety and also the efficacy profile of the investigational gene therapy being developed.
Okay, thanks you guys. You've been doing a great job submitting questions. We have another question about the stability of the GAA enzyme and the difference in the stability of that enzyme between the lysosome and in the blood.
So I think this is a really important and really good question. The principle of our gene therapy with delivering the GAA gene to the liver is going to still rely on one organ offering cross-correction to another organ. In other words, we still need for the GAA that's made by the liver to be secreted efficiently, and that's a large part of the bioengineering work that was done at Genethon, was to make a GAA that would leave the liver and get into the circulation well, but it still has to get to the organ that it needs to act. And in that respect, the efficacy or the usefulness of the gene therapy will have kind of parallels to how ERT, enzyme replacement therapy, works, in that it's still we're counting on a protein that's gotten into the circulation to make its way to skeletal muscle, heart, respiratory tissues, and to be able to enter those cells and work in those cells and in the lysosome of those cells. So the GAA that is made after liver-mediated gene therapy is not acting, is not having its functional effect in the bloodstream, and in this way it's a little bit different than, for instance, a gene therapy for hemophilia, where clotting factor in the bloodstream helps the blood to be able to clot, or gene therapy for diabetes, where insulin in the bloodstream helps the sugar control. In this case, we're still counting on the enzyme to, the GAA produced after gene therapy to get to, in particular, the muscle, and then work within the cell and within the lysosome at that site. The advantage, again, potentially, is that the engineered GAA we hope will be secreted from the liver in a reliable and efficient way and lead to a steady state expression of GAA in the plasma that does not have large peaks and troughs as one might experience after intravenous infusions.
And we received a couple of questions just about the clinical trial protocol. Again, that is not something that's been fully established at this stage as we continue in our preclinical research. However, once we have our clinical trials open and sites available, that will be posted on clinicaltrials.gov, and information will be provided to the advocacy community to make sure that you're aware of the progress ongoing at Spark. And I think I should just say to please stay in touch with us, as we do expect all of the energy around this program here to be moving us towards relatively frequent updates around how the program is progressing, and we look forward to continuing the conversation.
Thank you very much. Give me one moment. All right, thank you very much, Federico, Paul, and Amy. This concludes today's AMDA webinar. Thank you all for attending.
The moderator has ended the conference. Goodbye. Thank you for calling.